here with Jennifer Brophy, a leading researcher in the field of epigenetics at Emory University. She's here to explain to us a brief overview of epigenetics. Jenny, thank you for coming.
Thank you for having me, Sarah. So today I'd like to explain a little bit about what epigenetics is. So of all of the cells in our body, they all have the same DNA coding, but what makes a muscle cell different from, say, a nerve cell or a skin cell? Even though they have the same DNA, they act and look very different. So the way that this occurs is that as cells grow and mature, they receive different signals from other cells in the body or from the outside environment that leave little changes and modifications to the DNA. And these changes don't actually occur within the DNA coding, but in other ways. And these changes that don't affect the DNA code itself are what is epigenetics. So in addition to the actual DNA being methylated to open or close it for transcription, the histone proteins that the DNA is normally tightly wound around can be methylated as well to open or close the DNA and change the DNA's rate of transcription. So these are two ways that epigenetics occurs in our DNA. So these chemical tags, these methyl groups, are normally erased at both gametogenesis and embryogenesis, so when sex cells are made and when they come together to form a new organism. So when an organism is made, they don't have all the tags that their parents accumulated through their lifetimes from the environment and from their other cells. So sometimes the erasing mechanisms at gametogenesis and embryogenesis allow some of the chemical tags to pass through. This is what's known as transgenerational epigenetic inheritance.
We're here with Sarah Elliott, a Georgia legislator who's going to discuss proposed changes to the Georgia Performance Standards in biology.
Hi Jenny, thank you for having me. Today I'd like to discuss two different changes that have been proposed to be made to the Georgia Performance Standards in biology. The two different changes are located in two different sections of the performance standards. The first is Heredity Part 2E. Currently, this standard reads, gene mutations can be caused by such things as radiation and chemicals. When they occur in sex cells, the mutations can be passed on to offspring. If they occur in other cells, they can be passed on to descendant cells only. The experiences an organism has during its lifetime can affect its offspring only if the genes in its own sex cells are changed by the experience. The second standard that we propose to be changed is in the Evolution of Life Part 2E. It reads, new heritable characteristics can result from new combinations of existing genes or from mutations of genes in reproductive cells. Changes in other cells of an organism cannot be passed on to the next generation. We propose that these two statements are changed because we currently believe they are misleading. They imply that only changes to the DNA code itself can be passed on to offspring, and it is important to mention that not only changes to the DNA code, but also changes that are epigenetic in nature, such as histone modification and methylation of the DNA strand, can also be passed on to offspring as well. And it's critical to mention that these changes are important for evolution as well, and a lot of new studies have been going on, and actually epigenetics was founded around the 1970s, so it's a very established field, and we feel that there's enough research to support our claims, and that both of these two statements need to be amended to expand and include the definition of epigenetics as well. Thank you.
Sarah is a woman of many talents, and she is also the head researcher in a lab investigating the effects of epigenetic inheritance on evolution. Sarah, tell us how these two things are intertwined.
Sure. So as you can imagine, Jenny, the study of epigenetics has really evolved the way we look at evolution. A lot of new things have to be taken into account when looking and using our evolutionary models. For example, the rates of epigenetic mutation are actually much faster than the rates of mutation that are caused by changes to the DNA sequence. And that's mainly because things such as histone modification and epigenetic changes occur at a much faster rate. So as you can imagine, this impacts the way we have to look at evolution in the future. Moreover, our current population genetic models don't take into account certain effects that epigenetic changes have and some of the complexities associated with them. Epigenetics actually change what part of the DNA strand is being translated into proteins, but it requires proteins to make those epigenetic changes. So this sort of bidirectional change isn't being accounted for in our current population genetic models. Moreover, Epigenetics is actually just basically increasing the amount of phenotypic variation in the population that evolution is able to act upon. So because of this, we're looking at a wider amount of phenotypic variation that evolution is acting upon. So as you can imagine, all of these things are complexities that are changing the way the study of evolution is occurring. And while a lot of this is still speculative, we know that it is impacting evolution and that we'll have to continue to look at these changes in the future. Thank you.
The research possibilities are endless! So here we're going to investigate some of the new research that's being conducted on epigenetic inheritance and evolution. So first we'll take a look at Kumar et al. This study was conducted by the National Institute of Plant Genome Research, and in this study, Kumar et al. Was able to determine that there are a few sites of methylation in plants, specifically, that often the DNA methylation is often propagated to younger generations. So these sites of DNA are called the CG, CHG, and CHH elements. So Kumar et al. Was able to isolate these. They also found that in this type of plant called A. thaliana, A. thaliana has precursor cells to female gametes that are often mutant for methylation regulation, meaning that their female gametes, the clearing mechanisms that they have for epigenetic tags often don't work. So those tags are able to be passed on to younger generations. So they also cataloged a wide variety of experiments in which plants were given stresses such as high salinity and high temperature or bacterial infection. And then they observed the changes in the plant's progeny's phenotype and the amounts of methylation, and found that the environmental stresses often led to changes in methylation. So that's an example of these epigenetic tags changing and then being inherited in younger generations. So this poses the question of how epigenetic inheritance relates to Lamarckian evolution, because Lamarck proposed that changes during one individual's lifetime, responses to the environment would be able to be passed down to other individuals, and that because these traits were favorable, they would increase in population or in number in the population. So this is kind of like an example of Lamarckian evolution, and also Kumar et al. Raised the question of how epigenetics ties into agriculture if plants are given chemical treatments in the field, how they might become fitter due to changes in their epigenetic tags. Another study that we'll take a look at is Herrera et al. So these researchers decided to investigate the perennial herb H. boedatis and the differential transmission of methylation among different populations in different environments. So they looked at original plants and the seedlings of the plants and a range of elevations and forest types, and sequenced DNA and gathered leaf and pollen information in order to estimate the methylation amounts in both the original plants and the seedlings. They found that with the progeny, an increased seed mass corresponded with a decrease in methylation transmission, and that the plants in the lower elevation populations had an increase in transmission. So this raises the question, if methylation transmission is high, could that be a sign that a fitter epigenetic tag is being preserved, or vice versa? Or that perhaps with the struggle of adaptation that comes in a harsher environment, more tags are being passed on? These are questions that we have to look at.